Okay, so thanks for everyone coming on for another week of our weekly meetings. I'm gonna skip this because we usually have an idea for in-person meetings. Still, this can evolve, so people have ideas. Not any new papers yet in the past couple of months, but hopefully this one here in bioRxiv is under review. Might be accepted soon. We'll see. And then some updates again, just to update everyone that there's some case reports that we're working with Al Delaney, looking at interesting long COVID case reports. So if anyone's interested in joining us and working on these, let us know. There's definitely some help we can have, but there's already a good number of people helping us out. So if there's more people who want to help on this, it's an interesting case report. So let us know. Either email me or Al or both, and we could get you involved. All right, so let's just dive into the news. So I'm sure people have heard, again, I don't want to belabor the point about the origin stories, but, still in the news because recently things have come up again about, like the raccoon dog and other factors that keep popping up. But I thought if people are interested, there's a nice article that came out, a paper and a nice little article describing this paper in Nature, talking about the origins, about this recent paper that came out here just a couple of days ago, talking about it was from the CDC, Chinese CDC folks, their version of CDC, and they actually put out a paper looking at the swabs they did all around. Basically, they swabbed all around during the January 2020 and onwards about collecting samples and then sequencing it, and found out, yeah, indeed, there was SARS-CoV-2 positivity in the cages of some of these animals, like the raccoon dog and other species there. And as this article points out, and some of the other scientists, it suggests the possibility of animal hosts, and more strongly this paper. And there's some other paper by other folks, or other data coming out from other people who actually were there at the time and had done swabs and sequence. But saying this research, a lot of people also say the research still falls the definitive proof of, where is the animal-human spillover, but it's a good step forward. People were interested in. So it's a nice paper and it's a nice article on that. So now I guess people know about the couple of new variants that are evolving that are of concern. So I know we had discussions yesterday with a few people talking about the XBB.1.16 and then XBB.1.9. That should be in the next slide, that one. So I'll talk about XBB.1.6 first. But this is, people have been talking about the growth of the new spike, or the new, well, it's not really a spike, but the new growth increase in cases that we're seeing in India, and I'll talk about Singapore in a second. But this is because of the XBB.1.6, and although still low, but there is the increase that's happening in India that hasn't been seen in a while. And this in the U.S. we're starting to see some states that are starting to pop up, like California and these other states here. It's still low. It's like about, as I said, 3%, 2%, depending when you look up to, or 2 to 4%, depending on the region. This is around XBB.1.5 is still the dominant strain. But what the indications are, this XBB.1.16 is pretty close to the XBB.1.15. There's a preprint that came out from this Japanese group. It's a really small, it's a really minimal paper, but they just did a quick study to look at the effectiveness of XBB.1.6 and how, comparable to the other one, XBB.1.5, that's dominant in most places. But they see that, maybe this increase in uptake in cases is due to the effectiveness and the growth advantage it has due to some additional mutations that has occurred. And they're saying it's about 1.2, so 1.17-fold higher in reproductive rate than the XBB.1.5, which could account for these increases we see here. But I guess what they're saying, that immune evasion or the ability to have different impact on abusing the vaccines is comparable to XBB.1.5. So that, I guess, is positive news. And then it might account for the fact that there's not a rise in hospitalizations or ICU admissions yet, from what the WHO's reported in other places. So I guess take the good with the bad in this sense, but it doesn't seem to have increase in cases and people getting infected. So let me go to the next slide. So this is talking about XBB.1.9.1. So that's the other variant that's popping up. And the place it's starting to pop up is in Singapore. Here's a nice little web page. I never found this until now, coronaheadsup.com, that talks about various things happening with coronavirus and SARS-CoV-2 spikes. But what they see is, yeah, there's increases cases of hospitalizations and cases going up, minimal, but it's starting to go up there in Singapore. As you can see, due to the sequences, it accounts for 28% of the variants for the SARS-CoV-2 is XBB.1.9.1. Compared to the rest of the variants over there. And this is from, I took this from a Twitter post from Mike Honey, who's a data analyst, but he was looking at GESAID and looking at XBB.1.9.1 across the world. Like, for example, you can see the spike happening in Singapore. India tends to have this also for the variants that are being sequenced. You can see that India is starting to go up, but Singapore is the high, getting down here. Indonesia is actually the highest here. In Malaysia, and in the U.S., again, it's the low percentage, but you can see the states that are actually starting to, like California, Virginia, or Iowa, sorry, and other places in the East Coast are starting to pop. This variant started to pop up, and then when you look at the European and UK, you can see the places, again, low percentage of frequency happening, but it's starting to rise up in the counts. Again, as you see, the percentages here is between 1 to 4 or 6% of the sequences that are being coming in that are accounting for this. So it's something to keep an eye on and see what happens. I mean, some people are predicting this either XBB.1.16 or XBB.1.9.1 are going to be the next dominant strain that's going to come. But due to their, what we're seeing right now, really preliminary work showing that it's not going to be too different for, immune evasion compared to what we already have. So I guess we'll see what happens, how this plays out. I guess some good news. The FDA has now said it's okay to get a second Omicron booster for people who are at risk or have weak immune systems. So if and they're saying that that space is within the 4 months after after you got your first booster, you can be qualified again after 4 months to get a second one. But they say always consult your clinician or your doctor to see if you qualify or if it's recommended. But this is something that's been approved now, so if people wanted to get a second booster after the 6 months, this might be something to consider since it's been regulated now, approved. Some other, I guess, good news too. So this is talking about, this paper came out, so people might be interested in. I'm going to spend a couple of slides on this one. Talking about the a nasal vaccine that people are working on called SCPD9. And with these guys here, I think they're out of Germany, Berlin, they found that they actually has a better response than the standard mRNA vaccines like Pfizer, that compared to the Pfizer one, and it produced better serology, histology, and they did all their studies in the hamster, as you see in their little graphic here. But this is from their figure from their paper. The light blue is their vaccine, their nasal vaccine, and then the mRNAs that we know of that a lot of us have received is this pink one. But you can see overall, depending on what they're looking at, like whether it's the copies of the viruses or inflammation in the hamster, overall this vaccine seems to be producing a better response than the Pfizer vaccine, which is interesting to see. Another figure from this, they're looking at neutralization assays, and you can see this provides a better neutralization and antibody response. Again, the blue is the nasal vaccine. They did a combination too, mRNA plus the nasal, but mRNA plus mRNA, and then like a mock down here. But you can see that the nasal vaccine by itself does a pretty good job, a better job of antibody response and serology and neutralization, I should say. And they tested Omicron, Delta, and the other variants, earlier variants too. That's good to see. And then something that's of interest, is that they're looking at inflammatory markers, and I'm just showing you a few figures. There's more figures in the paper that you guys could dive into. But the inflammatory response that we see associated with SARS-CoV-2, it seems like this nasal vaccine is doing a pretty good job, if not better job than the other vaccines, to reduce that after the hamster's been infected. So that seems like a positive step forward for an alternative, easy to use vaccine. If it's a nasal vaccine, it might be something that might keep an eye out for the future when they actually go to clinical trials. Here's another paper that might be of interest to people. This is a case report, similar to the one we're working with Al, related to long COVID. That's a case report, but this one's related with how a mother could pass, before they talked about how a mom potentially could pass the virus to an infant when the mom is pregnant. I should say infant, but the fetus. And this is one of the first case reports that has come out from researchers in the University of Miami that show that indeed this woman who was two different women, this is a case report with two different women, who were pregnant at the time, that they actually did pass the virus to their baby that wasn't born yet, and this has caused brain injury to the infants after they were born. They found out. So they've done different than the case reports. So they have a few figures in here. This is one of them where they were able to take some histology and do some staining for different inflammatory interleukins. And they also looked at hypoxia markers in the infants. And you can see that indeed, like, the hypoxic events are being from when they're associated with the virus being infected, and then looking at different cases of the actual virus itself, in the placenta and other places, that they do find evidence that the infants, when they were still they haven't been born yet, I should say, that indeed that they were infected, and then this caused some impact on injury in their brain. What long-term damage, of course, is too early to tell, but that's something that has popped up. This is a nice little case report to show this. If you have any questions, just feel free to interrupt and ask. But yeah, so this was interesting to see, because I don't think people have hypothesized about the transfer of the virus from the woman to their the fetus, but this is probably the first time they actually show it definitively with some staining and some biology to show that it does happen. N of 2 again, case report of N of 2, so we'll see more people follow up with higher numbers. And then this was just the reminder that, even though the cases in the U.S. and in most places are very low, or getting down to, keep dropping. Of course, we have to wait to see what these new variants do. Does cause another rise in cases, but this is a letter that was in this JAM report that's saying, even though the cases are coming down, COVID patients, risk of death of COVID patients being hospitalized versus like the standard seasonal influenza in the fall and winter of 2022 to 2023, that, indeed, getting COVID, there's a higher risk of death as opposed to influenza. This is one of the tables and figures from the paper showing this. So this is more just the epidemiological study looking at this and comparing the deaths and the hospitalizations between influenza patients and COVID patients. And basically saying, maybe not a surprise to people here, but maybe the general public, to educate them to say, yeah, indeed, COVID has a higher risk of death still, even though you're vaccinated and comparing boosted and not compared to influenza. Here's some long COVID news. We're trucking along, so feel free to interrupt. This is not a paper, but this is a news article that I saw that might be of interest to people, and take this with a grain of salt, this news article, but there's some treatments that people are doing related to the impact of, loss of smell and taste that we know happens when people get COVID and get long COVID. So what they have been doing, this clinic in Cleveland and some other places, is that they do this stellate ganglion block. So basically, it's like a local anesthetic they do, which you might get, when you're at the dentist to get your cavities filled. But what they're doing is injecting that in the nerve cells in the person's both sides of the neck, and then they see actually some recovery from some of the cases that they have, or complete, recovery from their loss of sense of smell and taste. But that said, as they talk about in the article, you have to take this very cautiously because scientifically they don't know why this is happening. And there's a lot of unknowns here, so it's not recommended by a lot of people, but they're saying, I guess the article was highlighting that there seems to be benefits to this, so maybe people should find out why the benefits are occurring, and if it does benefit, overall cause more benefit than harm, then it might be useful for long COVID people. This might not be surprising, this news article here, but I thought I'd throw it up here that this study that came out, which is here in the Journal of General Internal Medicine, they did a study that's shown that for long COVID patients, 40% of them do have sleep issues. Again, might not be surprising for people here looking at that, but this is something that is talking about. And what's interesting, they said Black patients were significantly more likely to have this disturbance, maybe up to 3 times more than other races, which they don't talk about the biology again, but they saw this pure numbers, which was interesting to see. And then also people who have anxiety, greater anxieties had severity of the higher risk of sleep disturbances, which might make sense depending on what it is, how that's associated biologically. And here's they put out a press release here from Denver University from this group that was associating TBI might be, if you had TBI and then COVID, it would most likely worsen your response for long COVID symptoms. And I guess they put out a little preliminary results I guess we should wait until the paper comes out, but I thought if people are interested in reading what this lab here, the Linson lab, has done related to that, and so far what the data they have, it's not much, they don't talk about much here, but they're looking at different factors, drawn blood. I guess when the paper comes out, they'll be talking about some biological factors related with inflammation, astrocytes, and various other factors that could be contributing to TBI, contribution to long COVID symptoms, to worsening that. But I guess you could take a look at this article describing what they're doing and some preliminary results related to that. Here's an interesting paper that I think people might be interested in reading. It's a preprint, so again, take the preprint with a grain of salt, but I'm going to spend like a couple of slides here talking about this, or maybe just this slide here. But what's interesting is that basically what they found is that we've had other papers indicating that the virus might be remnant, the virus spike protein itself, or other parts of the virus might be remnant in the body after, long after you've recovered from COVID. This might contribute to long COVID. So this group here has found that the SARS-CoV-2 spike protein is actually found throughout the body, again confirming some other groups that have done the past few months. But they said that they particularly focused on here, the skull, meninges, and axis between, and they looked at both mouse and human models for this. So this is like the graphical abstract. So this is the brain, this is the meninges, skull, marrow, and then they basically found that, as they say in this graphical abstract, the SARS-CoV-2 and the spike protein is here. You can see it's been found throughout the brain. And this is some figures from that. So here's a figure I thought might be interesting for people to see. This is where they've done this both in the mice and the human. And the mouse figure, which is not here, they show that in multiple organs, including the heart and other places, this virus spike protein is found. Here they're looking at the brain and the meninges and the human skull, and they show the staining from the samples they've received. They see that the spike protein. You can see the arrows here, for example, is there between the meninges, brain cortex, and so on. You can see remnants of it throughout the brain. And what's interesting, they ran proteomics of, I'm sure I didn't check in this paper if they've made the proteomics data available publicly yet, but it probably should be sometime soon. They did it both in the human and mice. But with the axis, they're saying the main factor they think is attributing to this and causing downstream impact is PI3K and AKT signaling pathways they saw common between the mouse and human in the brain. That's something that they're touting, and they looked at different inflammatory markers, as a lot of people do. But PI3K, AKT makes sense to people here, since that's a pathway that's prominent for mitochondria. It's involved in mitochondria and inflammation and other factors too. Even a lot of different factors, even cancer, for example. This is a pathway that's found. but it's a pathway that people probably are familiar with. But they also show things like related hypoxia. You can see in this plot here that are being dominant between where these regions of the brain. What was interesting though, because whether the virus is replicating or not, they don't. I don't think they show that it's replicating. But what they did was they did an experiment where they took the spike protein or SARS-CoV-2, and they actually injected it in mice to see what might happen. And if in a mice that has not, in a mouse or a group of mice that have never been infected with the virus. So this is kind of simulating the remnants of the spike protein, as you can see in this graphical abstract, in the brain. And let's say if it persists, how it happens. Basically, in this mouse model, they show that by injecting just the spike protein in that region here, of the brain, in the skull marrow, it leads to brain cortex neuronal injury. They looked at different markers here. This is just part of the figure and discussion they had around data around this to look at it. But they looked at different markers related to this, including PI3K, AKT, and things like that. So that was interesting to see. Again, this is a preprint, so I'm sure things will change, but there's a pretty solid evidence of this. So something to keep an eye out, see what happens after the preprint. I don't know. People yeah, go ahead.
Sorry, just had a question. The human patients in this study, were they all people suffering from long COVID, or were some of them people who had come down with COVID but did not develop long COVID afterwards? I'm just wondering what.
Yeah, they were long-term. I think they were all long COVID. And I skimmed through this paper. I didn't see their methods, but that's a good question.
Okay, I was just wondering, because it's frankly perfectly possible that if you looked at the brain of someone who has had COVID but never developed long COVID symptoms, there might also be,
right,
fragments of protein floating around. It's just for some reason that person, isn't affected by them, or they're in such a low concentration that they don't have any bad consequences. I was curious whether they had looked at that comparison, but perhaps not.
I don't think they did, but again, I might have missed it. So I was looking at it this morning. I'll show the slides, but I would say from what I saw, I don't think they did that comparison, but that's a good point. Yeah. But yeah, take a look at the paper too. And I said, I might have missed that point, but from my quick read of the paper, it's your first point where it's people who are having the long-term neurological implications. They don't look at the acute one for the human.
Right. Okay, thanks.
Yeah. So I guess, maybe, this mouse experiment might address your point, Hannah, because this would be, it doesn't matter. These mice never had COVID or SARS-CoV-2 infection, I should say. So they just injected the protein, saying if this protein's there, it would cause neuronal injury. So this could attribute that, let's say, if what you're saying, like if someone does not have this long-term implication, someone is infected, and you still have this, this protein floating around your brain, it could lead to this neuronal injury, whether you know it or not, whether you're experiencing long COVID or long-term effects, neurological conditions.
The protein itself has a half-life of about 2 days, and so it's unlikely to be the protein that came from the original virus from months back, but it could be if there's some virus sequestered somewhere.
I didn't know that.
Or RNA sequestered somewhere.
Yeah, that's interesting.
Is that a predicted protein half-life, or is that a one that was actually determined? And where? How did they determine that?
That's a question for Eve.
Yeah, that's a question for me, and I'm not sure. I'll look.
Okay, yeah, no. Thanks.
Well, what you're saying is if a long COVID, if viral protein is detected in the brain or anywhere else of a long COVID patient, that protein is not a remnant of the original infection. That's what you're saying? Because the protein couldn't last long enough to be detected 3 months later.
Right. But there could be pockets of virus or something like that is what I was thinking.
Well, and the thing is that if you look at brain anomalies, so if you tau protein in normal cells, I don't think lasts months and years, but tau protein in plaques does. So if you have, if you have an abnormal brain brain physiology, you may get things that last a lot longer that don't normally. That don't, it really depends on the situation.
Like in Alzheimer's, presumably.
Presumably, yeah.
I'm going to quickly stop sharing so I could put this preprint link in the chat just in case people wanted to get it now. But yeah, no, sorry, continued discussion. I just wanted to interrupt. But yeah, I mean, they might have discussed this point, Eve or Sigrid or Hannah, but I don't know if they did. Because again, I skimmed through it. I didn't see anywhere talking about the half-life of the protein, so that's interesting. So I guess if you're a reviewer for this paper, you should bring it up if they have it.
Ashton, are there identification of the spike proteins specific to particular viral types, or are they general for coronavirus?
Well, it has to be, yeah. I mean, coronaviruses have their little spike in there, right? So other viruses not necessarily have the same machinery, depending what it is. And I don't know enough about other viruses to comment on that. Usually I refer to Nydia, but she can't make today's meeting. Maybe there's other viral experts here that know question the Bill's answer to Bill's question.
I'll write it down so I can research it.
Yeah. I mean, typically, we know with the coronaviruses they have the little spike, SARS-CoV-2 specific, right? And yeah, go ahead, Chris.
Just a comment about the topic. For other coronavirus, like pig coronavirus, for example, the spike protein of pig coronavirus is very different. So different that actually the target of that protein, the ligand, is in the intestines of pigs. So nothing to do with SARS-CoV-2 coronavirus. So I think it's very, very, very specific. And another comment about the presence of a protein there. I think that we saw other publications in which in long COVID there is like this reservoir of virus or antigen persistent, that is parts of RNAs of the virus is still there for some reason, and they are capable to be translated into peptides. So you can detect parts of nucleocapsid or spike protein, as far as I have seen.
Yeah, those papers I think came out late last year, right? Some of those papers you're talking about.
Yeah. It's last year papers. I remember something about it in the gut. Actually, it was full of detection of it wasn't dead people patients. Yes, I think so.
Did they say where it was located? Like, is there specific pockets of cells that it would be located in, or
I don't remember. Chris might remember. I don't.
It was. Yeah. They talk in these papers and discussion. They talk about some pockets of replication of virus that can persist, but it was an hypothesis. But because no one, and as far as I know, has been able to isolate a virus from there.
Right. That's what I've heard, and that's what in all our studies we don't see the RNA of the virus. But I could imagine there'd be little pockets somewhere, like other viruses that do that.
Sure. Yes.
Yeah, but definitely. I guess there's a lot of questions raised by this paper and others, but I think maybe see what you guys find. Yeah, sorry, go ahead.
Just only one observation of this paper. I checked the proteomics, and it's a very interesting upregulation of KRAS. That is a very important gene in cancer, related to cancer. It's a common duplication, actually.
Yeah, and that's something that I've hypothesized with the link, and I guess with Kashyap, we're going to discuss it more in the next months to come. So we'll potentially do experiments to prove one way or the other, if this is the case. But yeah, I didn't notice KRAS is there, but I know PI3K and AKT is involved with cancer pathways too. So that's interesting to know too. I didn't see KRAS there, but that's So yeah, if you see any other clues with any other things, Chris or others folks looking at the proteomics of this paper, feel free to reach out. So I'll be happy to collaborate in the ways we can. So Chris, is the proteomics available publicly? Is that what you found? Can we download that data? Because I didn't check it at all, just in case anyone's interested.
Not sure. Just look at the figures on the preprint.
Yeah. Okay. Yeah, sometimes in preprints they don't make the data publicly available until the paper is accepted. So I don't know. I haven't checked to see on this one, but if anyone's looking at it, if the proteomics are there, let's tackle it. All right. So here's another interesting article in Nature, talking as one of their articles, not papers, but discussing. I know we've discussed in the past the NIH Recover initiative, which is like $1 billion research program for long COVID, which there's a lot of critics. Some people here were critics that I don't think are here today, and with correct assumptions why you should be critical of it. But there was one thing that they were promoting. You might have seen this in the news, talking about exercise trials they're trying to do, saying it could be beneficial for long COVID patients, which might have some leg to it, but what they're saying more and more, as they discuss in this article with other scientists, that they're being criticized for that, because data that's been coming out is showing that since long COVID has obviously reports to the post-excursional malaise, the PM, the abbreviation for that, that after exercising, it's actually worsening the symptoms for a lot of people. Not everyone, but even light exercises. I'm sure if Bruce or Steve were here, they might agree to that when they were going through the thick of the things for long COVID. So it's an interesting article to read, the criticisms there, and also some recent findings of scientists talking about long COVID and exercise, and it might not be as beneficial as people are thinking, and people in the Recover program should probably reconsider how they're approaching it, and not waste funds, for example, as they're talking about. But just put that here in case people are interested in reading about it. Okay, I don't know if there's any other discussions for long COVID, but maybe I'll hop onto the variant distribution. We could always circle back. So this is
sorry.
Go ahead.
Could I interrupt before we move on?
Go ahead.
I just had I was just wondering, back to the pregnancy case report. Were the two women affected severely ill, or did they have mild cases? Or not that that's particularly, even a mild case would certainly be enough to make a pregnant woman nervous. But I was just wondering if this phenomenon of infection of the fetus is something that's more characteristic of severe COVID, or whether it could even occur in a mild case.
So from these patients, they were in the hospital when they were infected, so they'd be considered severe.
Right, bad enough to require hospitalization.
Yeah, exactly. So your mild question, we don't know. I guess that was the thing, since they were in the hospital, they were able to take samples and they consented, to that. So that was
right.
When you're mild, when you're home, no one's there to consent you for that. Unfortunately.
Right, I know. I don't know, if I were an obstetrician, I would be flipping out at this point.
Yeah, I agree. Yeah, definitely. I know there's been inklings and evidence. There were some cohort members previously, early on, who were, the specialty were pediatrics, and she was saying that indeed that the cases, there's evidence of it, not direct evidence, but the suspicions were there. There's enough evidence, indirect evidence, that she was concerned, but they had no data like this to show it, unfortunately, back then.
Hi, Ashton. Rebecca Lynn over at CHOP is a placental pathologist expert, and they're doing a study on placental. I think they have a paper or a preprint out.
Really?
Yeah, they're seeing massive changes in the placenta from COVID infection. So,
if you know what the preprint is, put it in the chat. I'll be interested to see that.
Okay, I'll try to find it. Yeah.
Thanks. Yeah. Yeah, that's interesting. I figure someone at CHOP will be looking into this, because that's what you guys do.
Yeah.
So.
And we're also partnered with UPenn's MFM, maternal fetal medicine group. So it's kind of a partnership. And
do you know if for Hannah's question, do you know if they're looking at all like severe to mild, or you don't know that?
They're just looking at placentas of women, of babies that are. I think they're looking at a lot of different people that have had COVID and had symptoms that were significant. I'll go find the paper.
Yeah. So maybe Hannah, Deanne has an answer to your question for this preprint.
Hopefully.
Yeah.
All right. Bye.
Thanks.
Thanks. Yeah, thanks.
Yeah. Ashton, paper came to my attention recently about loss of microbiome diversity, especially bifidobacterium in severe COVID-19. I posted that to BMJ in the chat.
Yeah, no, there's others who are looking at. I mean, one of our go back to the beginning. One of the papers released by our covert members late last year was discussing. Where's that paper? I have it here, somewhere here. Anyway, one of the papers that is related. Maybe I don't have it up here. I got to put it up here. Okay, so it's not up here. But they had looked into microbiome changes too. So there are people looking at that. And that's definitely a thing with this. But it's whether the microbiome is being a player along with SARS-CoV-2, or is this SARS-CoV-2 as a result of microbiome changes? Because any kind of disease always shifts the microbiome, and how that might play in, is I think people are looking into that. So, but is it? Yeah, that's definitely of interest to people.
I'll post the preventative stuff that I've seen too.
Yeah, we'll avoid that part until it's proven.
Okay.
Anyway. So I guess if there's any discussion more about other things, feel free to interrupt. We can always go back to the other slides. But yeah, so the variant distributions, again, as I said earlier, I mean, it's pretty much dominant XBB.1.5 as you see, but you can see in the US at least XBB.1.9.1, XBB.1.16 or 16 is still low, but it's around the 3 to 4% range popping up. California seems to have a higher range of it, so people in California should keep an eye out for XBB.1.16. Again, it's low, but see what happens. Despite of that, in the U.S., as wastewater monitoring nationally, it's basically still on the decline, so that's a positive note. And usually, as we know from the wastewater folks like Adam here and other folks who do this type of research, that this is indicative of what happens 2 weeks from now. So, in the population, you see a spike happening usually from the wastewater 2 weeks from now. So this is still in the decline, so that's positive news. It's still, if you look at this big spike here, this is Omicron, right? But this is starting to come down to potentially pre-Delta levels. It's not down to where we want to hope it would be zero, but if it ever gets there, we don't know.
I would ask Adam, do you see a decline in diversity in the wastewater samples of bacterial species present there, like bifidobacterium, before long COVID?
Bill, I'm actually off the top of my head, I'm not too sure. I'd have to kind of dig back into some of our historical data to really give a good answer one way or the other.
Okay, thanks.
And the wastewater is agreeing with everything else. The XBB is the dominant variant in wastewater, although they don't define. BioBot doesn't say if it's XBB.1.15 or other ones. I just show our county I live in Massachusetts. It's still declining. There's a couple of bumps that happen here and there, but it's still going down, which is good, positive news. And the variants from the CDC US tracker, you can see XBB.1.5 being dominant, but you can see this little dark blue color here is starting to be the XBB. What is it? Oh, this is .1.5.1. Which is pretty much the same as .1.5. I guess they haven't put .1.16 here based on the CDC called Duncan McConnell earlier this week. They haven't classified it yet, but it's going to be here, I guess, soon to find the variants. But you already see .1.9.1 here, the lighter, I guess what we call this blue color. This is navy blue. You can see that here in some regions. For example, the Midwest 7 region, there's a lot more of it than, let's say, East Coast. You can see California is here too. So it's creeping up, but on average right now, it's saying CDC tracker in the U.S. saying 5%. This version is the one that's spiking in Singapore. So this is the cases distribution. New cases, I think, is not very well or accurate. I don't show India here because India stopped reporting a while ago. And if you look on our world in data.org, it's all flat. There's no new cases at all in India, and that's unfortunate. It's just basically because of the lack of reporting and the lack of tracking which has occurred. But a better estimate or assessment of it would be, let's say, hospital admissions or ICU. As you see here, they're on the decline, which is good. France or Europe, you see that, again, cases, again, lack of reporting, but you can see a little slight increase in hospital admissions, but maybe because of the new variant that, XBB.1.16 that's in around Europe more, or .1.9.1. Either one of those, both of those are propping up soon. Germany, it looks like there's a decline still. US, we've already been over. South America and Brazil, there's virtually no new cases, but again, they don't report the hospital admissions and ICU, so we don't really know the accurate account to this. Sure, our Brazilian colleagues who are there and researching this can comment on what they see. They'd like. In Japan, this was the huge spike we saw in new cases, which they're doing actually a good job of monitoring it, better than other countries. This was the last Omicron wave that happened, but you can see now the cases are, and again, places like Japan and South Korea were like pretty model countries for a long time before Omicron hit them, because they were virtually very low in cases, and but then once Omicron took over, you got these huge spikes. But now they're back down. So I guess if Singapore is having their spike for .1.9.1, XBB.1.9.1, not sure how this is going to impact Japan. And here's Singapore. So this is the spike here. I guess this latest data, I kind of ignore this drop here. It's not accurate. It's noise, but you can see the spike actually in new cases occurring due to the viruses we've already went over. And this is it, I think, for today.